Most people don't realize it because they're invisible, but microbes make up about half of the Earth's biomass, whereas all animals only make up about 1/1000th of all the biomass. Craig Venter's achievements in genomic research broke barriers in the biological field by sequencing human genomes that help scientists detect disease causing mutations as well as creating fresh bacteria that can be used to break apart pollutants and make proteins for new vaccines. Since DNA was first sequenced in 1977, scientists have been trying to identify all 3 billion chemical units in the human genetic instruction set. The Human Genome Project, or HGP, officially began on October 1st, 1990, with the goal of sequencing the human genome and finding the genetic roots of diseases and then developing treatments. The HGP was the international research program whose goal was the complete mapping and understanding of human genes. The mapping of all the genes in the human body, collectively known as the genome, would lead to better medical management of diseases, as well as applications in waste control, environmental cleanup, and biotechnology. Diagnosis and detection of diseases and gene therapy could be included as effects of the sequenced human genome. The Human Genome Project Research Institute was the driving force behind the National Institute of Health, or the NIH, the largest biomedical research agency in the world. In March of 1999, large-scale sequencing of the human genome began. One man changed the face of biotechnology by surpassing the National Institute of Health, the largest single public founder of biomedical research in the world. Craig Venter, a California native, grew up with below average grades and no foreseeable potential as a successful scientist. When he was introduced to swimming towards the end of his high school career, he was just average. His coach gave him training sets, and he was so motivated that he spent 3 months of his summer training, never losing another race. He was headed to the Olympics until he was drafted to the Vietnam War. There, he served as a medic in a triage unit. It was his job to decide who deserved to live. Life was in his hands. The trauma of seeing hundreds of men die daily at the expense of war caused Venter to attempt suicide by swimming out as far into the ocean as he could until he was too exhausted to continue. Thankfully, Venter decided to turn back about a mile in. Swimming unlocked his potential, but Vietnam made him who he is. After he returned to the States, Venter went to a community college, then to the University of San Diego, where he initially wanted to be a doctor. After discovering science, he completed his PhD in physiology and pharmacology, became a professor at the State University of New York in 1976, and in 1984 joined the NIH, where he discovered thousands of human genes. The NIH patented them in Venter's name, which was unprecedented and upset many of Venter's colleagues who deemed him greedy. Venter left his job as a scientist at the NIH and broke off from the company to start a lab of his own. Venter's unique business model raised money from venture capitalists in return for Venter sharing his data with a for-profit company. In 1988, Venter officially founded Celera Genomics. It overtook the $3 billion HGP and mapped the genomes of the fruit fly and the mouse, both important laboratory animals. Venter used his extensive knowledge of genes to lead the first draft sequence of the human genome and assemble the first team to transfect a cell with a synthetic chromosome. In a competitive race with the NIH to be the first to sequence the human genome, Venter used the shotgun cloning method to duplicate genomic DNA. This technique was not founded by Venter, but was improved by him. Venter outperformed the billion-dollar national corporation and broke through the restraints they placed on him. He discovered a way to break up a genome into fragments using sound waves, and used a computer to reassemble it, rather than taking it apart one base pair at a time. This discovery allowed him to achieve his goal sooner than any other group could have because of his rapid approach. Riding on his success, Venter took a sabbatical and circumnavigated the globe. He undertook a scientific expedition around the world aboard his sailboat, Sorcerer II, and collected deep ocean samples so he could sequence their DNA and discover new life forms. Because of the information collected from his voyage, Venter sought to create the world's first man made bacterial cell. In 2005, he created his for profit company, Synthetic Genomics. In 2010, his lab made monumental history by creating the first ever synthetic life form.
We're here today to announce the first synthetic cell, a cell made by starting with the digital code in the computer, building the chromosome from 4 bottles of chemicals, assembling that chromosome in yeast, transplanting it into a recipient bacterial cell, and transforming that cell into a new bacterial species. So this is the first self-replicating species that we've had on the planet whose parent is a computer. It also is the first species to have its own website encoded in its genetic code.
Manufactured with chemicals in the laboratory and transfected with a man-made synthetic chromosome. His life form is self-replicating, meaning it can indefinitely make copies of itself. He created that bacteria cell by breaking the M. mycoides genome into 8 DNA segments. He then mixed and matched these to see which combination produced viable cells. Now, Venter has discovered that by changing the DNA in the cells changes the species by simply swapping out the genetic code like computer software. One of the very first benefits of this newfound synthetic life was the ability to create flu vaccines within a matter of hours rather than months. The synthetic DNA technologies synthesized the components for the vaccination within 12 hours.
Venter now believes he can uncover the deadly diseases lurking within seemingly healthy individuals with his new company, the Human Longevity Project. With human longevity, Venter hopes to detect diseases and make discoveries, not diagnoses. With his San Diego clinic, Health Nucleus, Venter will take DNA samples and use them to detect cancer, cardiac disease, metabolic disease, and neurodegenerative and neurovascular diseases. The genetic tests can cost upwards of $25,000, but the firm has found serious detections in roughly 40% of patients thus far, many of which were found much earlier than they would have been using traditional testing. Venter's genome was the first to be tested. After undergoing his own physical, Venter found he had prostate cancer, which has now been removed. Venter feels that the FDA has not yet found a way to serve the public using genome data regulation, so he is making his contribution himself. Venter also has found a way to prevent global warming with his discoveries. By eliminating the use of carbon and fossil fuels that deplete the ozone layer, the Earth will last much longer. His alternative to energy needs is the use of algae whose genomes have been altered. These algae would absorb the carbon in the atmosphere, then it would be converted into oil for car fuel.
Craig Venter's work with the Human Genome Project has allowed researchers to begin to understand the blueprint for building a person. By creating his microbe, Venter discovered a way to create proteins to be used in vaccinations, breaking barriers in the biological field. Venter's microbe allowed scientists to suck pollution out of the environment, slowing the depletion of the ozone layer, a process that is still viable today.